All right, thank you. So again, thank you all for joining us. This is one of what we hope to be many, what we call CAIRIBU Catalyst Conversations. And I'll just start out by introducing what CAIRIBU is. CAIRIBU is a consortium of NIDDK-funded centers and programs that are all investigating in some area or another benign genitourinary disorders and diseases. And it's right now it's about 16 centers or programs. The number fluctuates, of course, as the number of centers and programs changes, but we have U54 George M. O'Brien centers. These are kind of our flagship centers, which really brings together the thought leaders in areas of benign research. And then we have several P20 exploratory centers, and we also have 4 K-12 programs. 2 of these are K-12 programs dedicated to epidemiological investigations in benign urology research, and the other 2 are investigating are devoted to basic science and clinical research, translational research in benign urology. So we are, like I said, a consortium, very closely working with NIDDK program officers, and our main mission, we have several objectives, but the main mission is really to grow benign urology research and to increase the pipeline of investigators and to increase the diversity of those investigators working on various areas of the lower urinary tract. The other thing I want to say is just a real shout out to all the leaders of our CAIRIBU centers and programs who are joining us, as well as the trainees. One of our missions is to really, as I said before, bring forward new investigators into benign urology, whether that's from other fields or from the graduate school level onward. And I see many of our trainees on the call today. Our leaders have been very proactive in helping to provide guidance to them and mentoring. And this is really one of the main missions of CAIRIBU as well. So thank you all for joining us. I would now like to introduce Dr. Kathy Mendelson, who will be introducing Dr. Molly Ingersoll. Kathy Mendelson is a professor in the departments of urology and genetics and development at Columbia University. She is one of the PIs of the Columbia University George O'Brien Center. She is absolutely one of the thought leaders in our group, in CAIRIBU, as well as in urology more broadly. She earned a bachelor's degree at UMass Amherst in microbiology, got a PhD in microbiology at Columbia University, did a postdoc in Pierre Chambon's lab at the Pasteur Institute, a ton of publications. She's extremely well respected in her field, and I'm really grateful that she is in CAIRIBU. She provides so much leadership, and I'm really grateful for that. And I'm grateful she's here today to introduce Dr. Molly Ingersoll.
I want to introduce Molly Ingersoll, who I've known maybe for about 5 years and been incredibly impressed with her work. She's by far one of the leaders in the field of immunology and how it relates to the different organs in the lower urinary tract, for example. And she's one of the first people to kind of look into some of the issues that are kind of important. For example, males don't really get UTIs in the same way as females. Males get bladder, females appear to be protected in some way from bladder cancer compared to males. And these sex differences, some of them come down to immunology, and she's really paved the way in the field for this. And so I'm so happy to be able to have her join us this morning, and I hope we'll be able to find other ways to get you involved, Molly. And Molly's she's really right now at the Institut Cochin in Paris, but she also has an appointment, I guess, at the Institut Pasteur. And she's just moved to her new lab, and she's very happy. And I'm so glad to have you here today. Thanks.
Thank you both. I just want to say I'm really honored to be invited to something like this. I have had a passion for the bladder for a very long time, and it really means a lot to be recognized by an organization like this. And I'm always happy to share some of what we've been working on and how we think about the bladder. So I like to have a really sort of generic, broad dissecting immunity in the bladder, because I think there's always different open questions, and this really covers how we think about the bladder. So as Kathy mentioned, I was at the Institut Pasteur for the last 10 years. We've just recently moved our lab, but we're focusing on the same thing still. So we think about mucosal immunity specifically in the bladder, and that includes understanding the response to non-infectious inflammation, and this would be bladder cancer, for instance, how the bladder responds to the growth of tumors. It also is really therapeutic inflammation. So this would be, can we use immunotherapies? Can we use different types of drugs, including BCG for bladder cancer or checkpoint inhibition, things like that. And really more recently, and I won't have a chance to talk about it today, but we've been thinking about phage in the bladder for urinary tract infections. So there's a lot of really cool, interesting avenues to think about in terms of therapy. And what I want to talk to you about today really is our work in infectious inflammation. And so very simply, bladder infection, urinary tract infection. And I think given the audience, I don't want to dwell too much on, what is a urinary tract infection, just to say the majority of them are caused by uropathogenic E. coli, and that's the organism that we use for all of our studies, just because we think it makes sense to look at the predominant causative agent. We are all very well aware that this simple to treat infection is threatened by multi-drug resistance, antimicrobial resistance. I would say that the innate response has been well characterized. I don't think we know everything, but I would say there have been a lot of studies that focus on the innate response for many years now. And we have a really good idea about at least the innate response in female animals. And one of the things that keeps us up at night is that at least half of all women who've had one will have more than one infection. And so for us, these are questions that then relate to long-term or memory immune responses, and I'll come to that at the end. A majority of our work up until now has been using a really robust and I think very faithful animal model. And this is the reason we say this is we know that we can take human strains that have caused infection, we can put them directly into mice, and we cause an infection that looks very, very similar. It has the same clinical signs such as pain. We know that the transcriptional profiles of the bacteria are the same when they're isolated from human urine during infection or from mouse urine during experimental infection. That's the work of Harry Mobley, who likes to say it's the most expensive experiment they ever did. Which always makes me giggle. And mice can also have recurrent urinary tract infection. So we have the opportunity to understand what's happening at the level of memory response. So I keep coming back to this memory response, and that was really our first question when we started the lab. And my first PhD student asked the question, do we have memory after a first response? And so we use a really I think pretty simple experimental setup where we infect animals and we place a catheter in the urethra of the animal. We infect them, and then what we can do is we just simply count how many bacteria are present in the bladder after 1 day. And this is our baseline. So with E. coli, the strain that we use, we have about 10 to the 6, 10 to the 7 bacteria per bladder 24 hours after infection. And then what we do is we allow these animals to naturally resolve. So we don't do anything, we don't treat them with antibiotics, we just let them go. And what we do is we take urine every 2 to 3 days, and we just look for the presence of bacteria. And it's qualitative. This is just, is the mouse acutely affected or not? After about 4 weeks, all of our animals resolve. It's rare to have an animal that doesn't resolve. And when we have that situation, what we do is we challenge them. So we use the same strain of bacteria, but it's an isogenic strain in that the parents are the same, but the ones that we use have a different antibiotic resistance. And so that's how we can differentiate any residual bacteria from the first infection versus the second acute infection. And this is in part because of the work of Indira Mysarkar, who showed that reservoirs form in the tissue of the bladder after a first infection. So we infect the mice again, and we count how many bacteria are present. And what we see now is we have only about 10 to the 4 bacteria per bladder. And so this difference here, we assumed at the time was the result of a memory response. So the animal is remembering the bacteria, and now the response is stronger. And we support that with experiments in rag animals. So these mice don't have bacteria, or don't have B cells or T cells. And what we can see is the first infection and the second infection, they look the same. There's no protection. We can also do these experiments in animals that have been depleted of dendritic cells. And what we see is the level of protection is reduced compared to animals that were control treated. And we can also do this in animals where we deplete the T cells, and we see the same thing. So we know that we need components of the adaptive immune system, but we also know that this is a non-sterilizing immune response. So that means that there still is an infection, but there is a memory to it. So really we're trying to understand why. So the first thing we thought is we just need to look at the bladder itself. And just through some simple imaging, what we were able to see is that there are immune cells and they densely populate the bladder. So this is a transgenic mouse that we're expressing GFP from the fractalkine receptor, which is also called CX3CR1. And this is commonly found on macrophages and dendritic cells. So we thought, okay, we need to look a little bit more closely. We are immunologists first. So of course we have to do flow cytometry. And so essentially what we do is we take bladder tissue and we use a combination of enzymatic and manual digestion to separate the tissue into single cell suspensions. And then we just use a cocktail of different antibodies that will recognize surface proteins with different fluorophores. And so what I'm showing you here is a naive animal. This is a single bladder from one female mouse, uninfected. And what we're staining here with is CD11B, which is an integrin. It helps myeloid cells stick to the vasculature, and MHC class 2, which is a molecule that presents antigens, so commonly found on macrophages and dendritic cells. And this is what stimulates the development of an adaptive immune response. And so looking at this, you can see that we have a large population of cells up here that have both of those proteins on their surface. And when we look a little bit more closely using additional markers like this FC receptor CD64, what we can say is about 40% of all of these immune cells that are present in the bladder are macrophages. We also have dendritic cells, we have neutrophils, we have monocytes, but the macrophages really caught our attention because it's almost half of all the resident cells. So then we asked the question very simply, do macrophages influence the innate or the adaptive response? And to do that, we did exactly the same experimental setup that I just showed you. But first, we depleted all the macrophages in the animal using an antibody. And we tested to be sure that this really removes the macrophages from the bladder tissue as well, which is really important because the penetrance of different antibodies is different in different tissues. We're really surprised to see that taking away those macrophages and infecting the animal 24 hours later, there is no difference in the number of bacteria present in the animal. This is surprising because we do see that the macrophages eat the bacteria during infection, but it seems like they're not the primary cell to clear the bacteria. But so we went on and we did that same waiting for 3 to 4 weeks, and we challenged the animals. And what we saw, quite surprisingly, is the animals that had been depleted before that first experiment were better protected than the control treated animals or the completely untreated animals. So this is just the challenge right now, which you're seeing on this graph on the right. And what's important to know is those resident macrophages that we depleted, they came back. They were present during the second infection, but somehow something had changed. So their absence in the first 7 days or so changes the immune response, and the animals are actually better protected. So that tells us that macrophages, resident macrophages, are somehow impairing the development of memory. And so we can sort of visualize that here. We have bacteria that'll infect, they cause apoptosis in the urothelial cell layer. That leads to the release of cytokines, which ultimately recruits neutrophils and monocytes, which are really the cells that are clearing the bacteria. But we have these resident macrophages that somehow are impairing this memory response. So of course, we got really interested in these macrophages, and we went a little bit more, looked into their biology a little bit more deeply. And what we saw is, again, using that same protein marker, CX3CR1, which is, like I said, a chemokine receptor, we saw there are actually two kinds of macrophages present in the tissue, which was really quite surprising to us. Looking back, it shouldn't have been, because a lot of different tissues have different types of macrophages, but we didn't expect that to be the case in the bladder. We went on and we were able to measure other types of proteins on the surface. So LIVE1, for instance, which is a molecule that these cells use to associate with endothelium and other vessels, and TIM4, which is a receptor for dying cells. And so these molecules suggested to us that perhaps these cells actually have different roles in the bladder. And so we looked a little bit more closely at them, and using particularly LIVE1, which is great for imaging, we could see that macrophages were in different locations in the bladder. And so we found one type of macrophage that was predominantly in the muscle. And we decided to name those MacM macrophages from muscle. And then we found a second subset, which you can see here in this image, all here in red, that are predominantly found in the lamina propria, very close to the surface of the bladder, in fact, because all of this dark space is the lumen of the bladder, and this pale blue are the epithelial cells. And so those ones that are in the lamina propria, we call MacL macrophages. So now we have two subsets, and we really wanted to understand if they're actually different from each other. So we did RNA-seq on these two populations sorted from naive animals. So again, this is all uninfected female mice. And what we can see here is that there are a total of about 1,500 genes that are differentially expressed between the two subsets. And when we went a little bit more, sort of deeper into the data, what we saw were a number of different phenotypes. And I'm showing two examples here. Phagocytosis, for instance, which looks like there's differential expression between the two subsets, suggesting that the MacM macrophages maybe are more phagocytic, and MacL macrophages had really high levels of genes associated with apoptosis. So these are phenotypes that we can test in our model, which is really fantastic. So what we did is we infected mice. And so I'm showing you the same type of analysis where we're looking at all of the different cells that are present in the bladder. Now this is infected animals. And what we can see here, this large population is actually monocytes that infiltrate. So this is the response to infection. But up in this top gate, those are our resident macrophages. And so we can take those we take that resident population, we can split it into those two subsets using, again, this exact same protein chemokine receptor. So we see our two types. And then what we can do using a uropathogenic E. coli that expresses RFP, we can ask what cells have the bacteria inside of them. And then we can quantify that. And what we see is those MacM macrophages in total, more of them have bacteria present inside them compared to the MacL. And this is sort of curious because the MacM macrophages are in the muscle. They're farther away physically from the infection compared to the MacL. And so we don't know right now how these MacM macrophages are actually taking up bacteria. And we don't know if it's direct or if it's because they've taken up infected cells, for example. And so we can look at total percentage of macrophages that are infected. And if we do it by a percentage of where the bacteria are distributed, you can see that a majority of them are in that MacM subset. So moving on to apoptosis. One of the things that we noticed quite early on is about 24 hours post-infection, we see, where's my mouse? Here we go. We see that the number of MacL macrophages is lower than it is in the steady state. We do have a difference now. There's more MacM compared to MacL, and we know comparing it to the steady state, to the naive animals, we know that this is a reduction in MacL and not an increase in the MacM. And if we look now at the proportions, we can see again that there's many more MacM compared to MacL, whereas in the naive uninfected state, it's about 50-50. So we decided to look a little bit earlier, and we can see that we already have cell death as early as 4 hours post-infection. And that again impacts the percentage of the two subsets. And we were curious about why this is. And my PhD was actually studying Shigella, and Shigella induces cell death in macrophages. And so we thought, okay, infected cells must be dying. But what we did is we looked at the number of dying macrophages that were in total, and we can see that the number of dying cells, this is using a cell death marker, is higher in MacL macrophages. But if we look at how many of those are infected, there is no difference in the number of infected dying cells between the two subsets. So we don't think that it's a direct mechanism of being infected that's inducing death, but we also don't know what the mechanism is at this point. So we were really quite curious. Now we have these two macrophages in two different places that are transcriptionally diverse, where one dies, one of them takes up more bacteria, but we were curious if they had different roles in terms of the rest of the immune response, knowing that their presence is detrimental at the beginning of an infection. So what we did is we actually did a little bit of a swap, and instead of doing the depletion before the first infection, we did the depletion before the second infection. So the mice were all infected at the same time. We took that 24-hour time point, we waited 4 weeks, and then we depleted macrophages before the challenge. And so what I'm showing you here first is that when we deplete those macrophages before the challenge, the mice, again, are better protected compared to the control scenario. They have fewer bacteria present in the tissue. We reason that maybe it's because these macrophages are responsible for recruiting other immune cells. That's been shown in other systems and also in the bladder. But we saw no impact on recruitment of, I'm just showing you as an example here, neutrophils, but also on eosinophils or monocytes. So we don't think that this protection is due to an enhanced recruitment of immune cells, for example. And so what we found instead, which was really quite interesting, is in the absence of these macrophages, we see a change in the polarity of the immune response. So essentially we see more T cells that are biased towards the T helper 1 phenotype, and T helper 1 T cells are important for fighting against intracellular infection. So this is not super surprising, but this was really quite remarkable that we see the skewing of Th1 T cells and other effector cells, NK T cells and NK cells, which are all important to fight the same type of intracellular infection. So somehow taking away those macrophages creates an environment where we can have an alteration in the microenvironment or the immune polarization of the bladder. So just kind of to sum up that first part, essentially what we understand is that there are two spatially distinct macrophage subsets. I'm showing them, I'm representing them here. And they play different roles during urinary tract infection. The one that's closer to the lumen somehow dies very early. We don't understand why during infection. And the second one is able to take up more bacteria. We know, and I'm not showing you the data today, that infiltrating monocytes can come in and they can differentiate into both types, MacM and MacL macrophages. We need to know that these are transcriptionally distinct from their naive counterparts. And we know from our depletion studies that they also seem to negatively impact the development of a protective immune response. So our macrophage studies are absolutely ongoing. We're trying to understand exactly what's going on in terms of bacteria uptake or in terms of cell death. But we also asked the question in the lab, what other factors influence immunity? So Kathy mentioned I think this is my favorite aspect of our studies, which is understanding the influence of sex or sex as a biological variable in all of these different bladder diseases. So I'm just showing you here, the circle on the outside in gray is meant to represent the number of these different types of bladder diseases. So you can see this very dark gray is the number of urinary tract infections that happen per year in the United States. The number is somewhere around 8 million. And we have this really small piece here, which represents bladder cancer, and then, of course, catheter-associated infections. And as we move inside the circle, the red is meant to represent the number of women who experience these bladder diseases compared to the number of men. And as you'd expect, catheter-associated infections really don't have a sex bias. You put that catheter in, for us, it's a lot like you're inoculating the patient, and so you're going to get an infection. Bladder cancer is really quite interesting because about 75% of all patients are men. And we do know that there's a role for androgens and androgen signaling in the development of these cancers. And then, all right, here, just to stick with our theme of urinary tract infection, you can see this really big red circle with just this tiny little cap of blue. And that represents the fact that women are about 40 times more likely to experience an infection during adulthood compared to men. But that's not to say that urinary tract infection is limited to women. And I think this is a really important point to make, even within this community, is what I call the urethra hypothesis, which I think we want to get away from. And I'm happy to see Kathy shaking her head. The scenario we think about quite often is this: adolescents and adults, we see infection in women, it's much more common, but we can say roughly 25% of the entire population experiences infection. And then the size of this, these two symbols is meant to represent the prevalence between the two sexes. But if we consider the two extremes of life, infants, for example, we know that infections are a little bit more common in baby boys compared to baby girls. And overall, about 10% of all infants will experience infection. And on the other end of the spectrum, which is really quite interesting to us, is that adults over the age of 50, 55 or so, the prevalence of the incidence changes again. About 10% of the entire population will experience UTI at some point. But what's interesting is the prevalence in men increases dramatically and seems to decrease in women, so it's about equal between the two. So we think, oh, and the really key important point here is there's actually a really nice study that shows the urethra length does not change between these two populations. And so that's really not an explanation then for, or at least not the sole explanation for why we have differences in this period of life. And so again, we're immunologists, we're a little bit biased. So we think that the sex bias in UTI is actually due to differential immune responses to infection. And to explore that, the first thing we did is we just optimized how to do infections in male and female mice. And so we used the classic model that everyone uses, which is a catheter into the urethra for female mice. And we just adapted the same thing for male mice. If anyone's interested in this, we published a Jove a few years ago where you can see a video of us and we explain exactly how to do this. And again, if you're really curious, you can reach out. We've taught a number of labs how to do this. So what we did first is we infected the animals and we just looked to see who's infected over time. Very straightforward experiment. And I like to tell the story that this happened over Thanksgiving, and I was out of town and Anna wasn't sure what to do. So she just took urine for a month. So that was great. And essentially what we can see here on the y-axis is that we're just counting the number of mice that are infected, and you can see over time that the number of infected female mice diminishes until we get to 0, whereas in male mice, we see anywhere from 50 to 100% of the mice remain infected over time. The longest we've gone out to is 42 days. And so we look a little bit more closely and we count how many bacteria are present. This is the same experiment that I've already shown you in a number of different ways. We can see that this difference is not because they're differentially infected in the beginning. The number of bacteria present at day 1, day 2 is not different between the sexes. It's not until we start getting later in the infection that we start to see that some of these mice remain chronically infected, and ultimately at day 28, all of these male mice still have bacteria in their urine, and all of these female mice have sterile urine. So yeah, they are still colonized. Again, this is this tissue reservoir, but none of these mice have an active infection. So our first guess was that the reason we have this difference is there must be a difference in the adaptive immune response. And so we did exactly the same experiment I showed you already, which is to infect, to take a 24-hour time point, which is always the first one. This is 24 hours in a female mouse, 24 hours in a male mouse, and then to wait 28 days. The only difference here is we have to treat the animals with antibiotics at day 28, because the male mice will not clear, they won't resolve their infection on their own. We treat the female mice as well, just to account for any any impact of the antibiotics, but we don't see a difference between antibiotic treated and non-treated female mice in terms of their response. We also don't see a difference in the level of protection between male mice and female mice. And so very early on in this project, we were able to rule out that protection or the development of memory is not different between male and female mice. And so we turned then to focus more on the innate response. And so one of the things that we like to do when we are doing bladder CFU, when we're counting how many bladder are present, we can use those same samples to measure cytokines that are present. And so what you're looking at here, it's called a spider plot. And these are all of the different cytokines and chemokines that we measure in 24-hour infected animals. And this is a log scale, which is important to point out, because then you can appreciate that this red line, which is the level of cytokines present in female, is much higher than what we see in male bladders. And in some cases, you can see that there are some really big differences. We also compared this to animals that had been instilled with just PBS to account for any potential damage that's induced by the catheterization itself. And so what we see at 24 hours is there is a big difference in the cytokine response, but interestingly, that difference is gone by 48 hours. And so essentially what you see is male and female lines almost completely overlap with each other, but they also almost completely overlap with this dotted line, which is a completely naive animal that's not even been catheterized. And so what this says to us is that immune response is strong and it's acute. It happens in the first 24 hours. So whatever that difference is, it's in that first 24 hours. So before we went on with this, of course, we wanted to make sure that, excuse me, we weren't doing some, we weren't following up some anomaly that appears only in mice. So I'm really lucky to, up until 6 weeks ago when we moved, to share a lab and an office space with Dr. Dara Duffy, who co-runs something called the milieu interior. And so the milieu interior is a collection of 1,000 individuals, all healthy, half men, half women, aged 20 to 69. And one of the measurements they make is using these devices called true culture tubes, which I'm showing you here. So essentially it has a proprietary medium in it, and you can put any type of stimulus you want. This is a list of what's present in the milieu interior. And then what you do is you take blood from these healthy donors, you put it in, and from that, you can measure in the plasma, you can measure cytokine responses, and in the cell palette, ultimately, you can measure transcriptomics. And so we were really curious about some of the responses to this. And so this is just a representation of one of the analyses that they did, which is published here in PNAS a few years ago, where this is comparing the male and the female donors, the non-stimulated tube, to the tubes that contained E. coli, Staph aureus, BCG, Candida albicans, influenza, and the superantigen here. And essentially what they did is they looked at the expression level of about 560 different immune-related genes. And from that as well, they have imputed SNPs and they can look at cell populations. But we're really curious about these induced immune genes. So what we did is we looked at a subset of their data. We looked at the induced immune response following stimulation with E. coli. And essentially what I'm showing you is this is a PCA plot of these 560 different genes in donors that were between the ages of 20 and 50, because we're really interested in that period when women get many more infections compared to men. And we wanted to remove the additional layer of complexity with menopause. And so what we did is we overlaid the sex of all the donors on this plot. And what you can see is that naturally men and women pull apart. You can see the female donors separate from the male donors. So we were excited by that because that suggests that there is a sex difference in the immune response to E. coli. And then on top of that, what we did is we overlaid the level of expression of a few genes that we had the data for in the mouse model. So in our mouse model, we can measure CXCL2, IL-8, or the ortholog of IL-8, of course, LIF and IL-1. And what we can see in red are genes that is a higher expression, and green is a lower expression. And you can see that overall the pattern is the higher expression is coming from the female donors, whereas the lower expression is coming from the male donors. So this really gave us some confidence that what we were seeing might be biologically relevant in human infection. So we went back to our animal model and we asked, okay, what's the consequence of having this differential immune response? What's the consequence of having these different cells, different cytokine expression levels? And so what we did is we measured immune cell infiltration. So what I'm showing you here in red is always female mice and blue is male mice. And now we're looking at the number of cells, again, using flow cytometry. So I always have ×0. These are naive mice. These are uninfected. And so we can see if there's any differences ultimately just between male and female animals. And then again, these are all on a log scale. And what we see at 24 hours is there's a greater number of total immune cells in the bladder of female mice compared to male mice. And those differences disappear at 48 hours, just like the cytokines. We see that these resident macrophages I just told you about, their numbers don't really change. They do change from naive, but they don't change between the sexes. We see a tiny bump up in the dendritic cells. And that sort of correlates with the fact that we don't see any huge differences in the adaptive immune response between the two. We do see, however, big differences in the number of neutrophils, monocytes, and eosinophils that infiltrate the bladder. Now, I told you it's our work and many other people that neutrophils and monocytes are really the cells that are clearing the bacteria. So this gives us a clue as to why female mice have may possibly have a better resolution. And again, eosinophils, anyone who's interested or has an idea, let us know. We don't know what they're doing in the tissue, but there's plenty of them. And I think it's an interesting question. What we also are able to do, again, using, click, there we go. Using these red bacteria in flow cytometry is we know that the bacterial uptake in the cells that are present in the bladder is different between female and male mice as well. So if we aren't counting the number of neutrophils, but just looking at neutrophils as a population, a higher percentage of them contain bacteria in female mice compared to male mice. So that's, again, an open question for us because it suggests that there's differences in phagocytosis between the sexes. So we really started trying to understand, so why is this, what's going on? And if we take the graphic that I showed you earlier with the percentage of the population impacted and the distribution in terms of sex and who's impacted, we can put that underneath sort of this timeline of hormone expression in humans. And what we see here is there's this teeny little burst of testosterone shortly after birth in male babies. But ultimately, hormone levels in general are really quite low until puberty. And then what we see is very high levels of testosterone expressed in men and very high levels of estrogen in women and lower levels of the two in the opposite sex. And it's only after about the age 50 or so that we see a slow decline of testosterone in men and a really rapid decline of estrogen in women. And so these differences suggest to us that the immune responses we were seeing might somehow be modulated by hormones. And so we amended our hypothesis, if you will, and we think that the sex bias is actually influenced by sex hormones. And so what we did is a really straightforward approach, which is just basically taking them away or adding them back to the system. And so in male mice, we can castrate them, and that takes away testosterone. And so what I'm showing you here is just a measurement of testosterone levels. These are naive female mice. They have very low levels. These are naive male mice, and this has been reported previously that within a cage, you're going to have a really wide range of testosterone levels. There's always that one dominant mouse who's pulled the whiskers out of all the other mice in the cage. So you know which one has the highest level of testosterone. When we castrate the animals, we can get those levels down to almost really to the level of female mice. We still once in a while have one or two outliers. These guys get taken out of the experiment. And then in female mice, to add testosterone to the system, we use something called T-tubes. And these are essentially sialastic tubes that will slowly, when wet, will slowly leak out whatever you put inside of them. So we make them ourselves, and we essentially glue one end, pack it full of testosterone, glue the other end, and implant it under the skin. And these can last for about 3-4 weeks in these animals. And with that, we can get a uniform, physiologically relevant high level of testosterone in our female mice. And we do a mock surgery with empty tubes as a control. And so essentially what we can see when we remove testosterone, the male, it doesn't change anything. The male mice remain infected for 1 month. And ultimately what we see is the bacterial levels are the same. In female mice, what we see when we add testosterone is now they can no longer resolve their infection. So it's really, really exciting to us to see that with just the addition of testosterone, we can now completely ablate the animal's ability to resolve their infection. And what we see is now these levels of bacteria are really quite high. But we still didn't have a good idea what's mediating resolution, because when we looked at the immune cells, there weren't really any differences. There were some differences in cytokines, but almost no differences in these myeloid cells, neutrophils, monocytes. So we thought, okay, we have this scenario where male mice don't resolve. We have this scenario where we add testosterone, female mice don't resolve. And we had this other scenario in the lab, which was a different project, where these rag gamma C mice can't resolve their infection. This is a little bit unfair because a rag gamma C mouse is missing B cells and T cells and NK cells and gamma delta T cells and a number of different cells. But it gave us an idea as what we wanted to look at. So we shifted our attention away from myeloid cells, away from monocytes and neutrophils, and more towards lymphocytes. And so we looked at the number of CD4 positive T cells, NK cells, gamma delta T cells, and a subset of innate lymphoid cells called ILC3s. And so these are cells that are a little bit comparable to the Th1, Th2, Th17 paradigm for T cells, but they're innate. And essentially what we saw is, with the exception of gamma delta T cells, there's really no differences in naive animals, but what we saw is all of these populations increased only in female mice during infection and not in male mice. And when we gave testosterone, those increases were reduced compared to untreated mice. And with the exception of CD4 positive T cells, with the exception of NK cells, all of these cells make the cytokine IL-17. And if you were squinting at your computer, you might have seen that the biggest difference at 24 hours is in fact in IL-17, where we have a log and a half, almost 2 logs difference in the level of IL-17 that's expressed. So we really went after IL-17. And what we did is we used an antibody to essentially neutralize this cytokine at the time of infection. And what we saw is when we take away IL-17 in female mice, they lose the capacity to resolve their infection. And so ultimately what we know is that IL-17 is absolutely necessary, but when we put it into male mice, it's not sufficient. And we think that's because in addition to missing IL-17, they're missing a whole bunch of other cytokines. So they're really missing that whole inflammatory milieu. But what we can say is in female mice, we have this distinct cytokine signature, which is here. So we have all these different cytokines. They recruit immune cells, they activate the macrophages, and we have this robust adaptive response that happens afterwards. In a male mouse, what we have is we're missing exactly this. We're missing this cytokine response, and that leads to a lack of cells infiltrating, and ultimately we have chronic infection. And so from this, we can say that we know macrophages modulate the development of adaptive immunity. We know that pronounced inflammation that includes, and absolutely it's necessary to have IL-17, is needed to resolve UTI. And then if I have just 2-3 more minutes, I want to show you some brand new data. We just put this data up on bioRxiv maybe a week and a half ago. We're really, really curious to understand what factors are ultimately influencing memory in the first place. So I told you in the beginning that we determined that adaptive immunity is developing in the bladder because when we infect mice that don't have T cells or B cells, they don't develop a memory response. So we wanted to get a little bit more into that. So the first thing we did is this is always going to be the control scenario on the left. So this is the first infection, and this is the second infection in the open circles. And you can see there is this non-sterilizing decrease in bacterial burden. So that's our protection. First, we did this infection in mu-MT mice. So these mice don't have mature B cells. So essentially you can't make most antibodies. And we're really quite surprised to see that these mice were protected. It didn't matter. There was no difference in the level of protection. This is a little bit surprising because the literature is a bit confusing on this, but there's definitely papers that suggest that B cells and antibodies are protective. And in fact, the presence of antibodies in the urine is suggestive of a urinary tract infection. So maybe in this particular scenario, no. We then went on to deplete just CD4 positive T cells, and we were really surprised to see, again, these mice are still protected. And we depleted CD8 positive T cells, and again, the mice were protected. So from this, what we thought was maybe it's CD4 or CD8, and one can compensate for the other. And we ultimately decided to focus on CD4 T cells because there's many, many, many more CD4 T cells in the bladder after infection compared to CD8. So now we have to get into T cell biology, and this was a bit of a new space for us. So we had to do some basic reading, but essentially there's different T helper subsets. So I mentioned this T helper 1 already. It's important for bacterial infections. T helper 2 is generally associated with the response to allergy or parasite infections. And T helper 17 is a really pro-inflammatory response also associated with a bacterial infection, but also really, really well associated with Candida infections and fungal infections in general. And so the first thing we did is we just wanted to understand what T helper subset was there. What's the polarization? What's happening in the bladder? And the reason we looked at that is because a recent study suggested that a T helper 2 response was predominant, and that's why the immune response was not sterilizing. But there were 2 caveats to this study for us. The first is they only looked in female mice, and we already had published evidence that T helper 2, that type 2 immune response, doesn't actually happen in male mice, but they are still protected. So that suggests the skewing is not what's mediating this. And in this study, they also neglected to look at IL-17 or Th17 T cells, and we know that IL-17 is absolutely key for this response. So this is a lot of data, but the point here essentially is we looked at the T cell bias over time from day 0 all the way to day 7 in primary infected and challenge infected mice. And what I mean to show you here is this red dotted line is 50%. So there is no Th1, Th2, or Th17 population that's predominant. Okay, so we get something called a mixed TH response. And we think that this is essentially reflecting the complex lifestyle of the E. coli in the first place, because they're both intracellular and extracellular. So what we saw is there was no particular response. And we also saw that there was a high number of regulatory T cells, which we know will impact how memory develops. So we thought, okay, we have this scenario that polarization is maybe not the determining factor. And we have this T regulatory scenario suggesting that we might have something like tolerance in the bladder to infection. And so what we did instead is we wanted to play with how much antigen was present. And the reason for that is too much antigen or not enough antigen will lead to tolerance instead of memory. So we wanted to test if that was the case. So previously we had looked at increasing amounts of bacteria and we saw no differences in memory. So we went the other way this time. And so this is the work of Mathieu Rousseau, who's been a postdoc in the lab for about 5 years now. And what he did is he infected mice with either 10 to the 7 CFU, which is what we always do, or 10 to the 5 or 10 to the 3. And what you can see here is the animals with the lower inoculum resolve their infection faster. That's not surprising. And again, this is the control scenario. And we see when we give fewer bacteria, we no longer have memory. And when we give 10 to the 3, we see that the second infection, which is always going to be 10 to the 7, looks like the animals, looks like a first infection. So, okay, we know that there's a level of antigen that's needed for memory. We wanted to recapitulate that in a bit more of a clinical scenario. So what we did is we gave antibiotics to mice at day 1, day 2, day 3, or day 5. And we're really surprised and I think a bit disturbed even to see that when we give antibiotics at day 1 post-infection, we lose protection. These mice no longer generate a memory response to infection when they receive antibiotics in the first 24 hours. And what we were able to show, and I won't show you the data because it's not different, is T cell polarization doesn't change under any of these conditions. So we thought, okay, it's not T cell polarization, for sure, not, what is it? And so now we have to do some more T cell biology, which is to understand the types of memory that develop. So there's central memory. These are long-lived cells that live in the lymph node. There's effector memory. These are, again, long-lived cells that circulate. And then there's something that was more recently described, which are tissue-resident memory T cells. And there's only 2 reports that they might be present in bladder cancer and no reports related to infection, and very few reports related to bacterial infection in general in any organ. And so we went in and we looked to see if those were there. And essentially what we found is in this scenario where we gave only 10 to the 3 bacteria instead of 10 to the 7, and when we treated with antibiotics at day 1, we have a reduction in the number of tissue resident memory T cells. And the cells are special because they're already in the bladder. They're poised to respond to infection very, very quickly. And so now we see a reduction in them in the same situation where we don't have memory anymore. And so we needed to understand if they were necessary. And so we took advantage of what I told you in the beginning, which is different antibodies have different penetrance into different tissues. We treated mice to deplete them of CD4 and CD8 T cells. And what you can see here is we can deplete these cells. I'm only showing you CD4 for right now, but we can deplete them in circulation. We can deplete them in the lymph node. We can deplete other types of T cells, but in the bladder, we can't deplete these cells because the antibodies don't reach these cells. And we were able to show that it's not a masking issue, but really these cells are not touched. So now we have a scenario where the only memory cells that are present in this mouse are in the bladder, nothing in the lymph node, nothing in circulation. And we did the exact same experiment where we infected and then we treat it either with isotype or with antibodies. And what we see in a mouse that only has tissue resident memory T cells, we have the same level of protection as our untreated animals. And so from this, I think we can say that this tissue resident population is necessary and sufficient for protection against a second infection. And so essentially that leaves us with tissue resident memory cells mediate adaptive immunity. But it leaves some clinical questions, which are we setting patients up for recurrent UTI by giving rabbit antibiotic treatment? Can we use combination therapy to offset the impact of antibiotics on memory? Or can we develop a vaccine that will directly target the development of tissue-resident memory cells? And that's a really active area of research right now in other mucosal organs. And then more fundamentally for us, of course, we're super curious about how our macrophage subsets and our T cell subsets are interacting in the bladder. And we're now doing some work in bladder cancer in human samples, but also continuing our mouse studies. And we're really curious about what factors influence all of these interactions. And so just with that, I really want to point out Mathieu Rousseau did a majority of this work along with Olivia Lasser de Mariano, who was an amazing PhD student. And this is my old team at Pasteur, and this is my intermediate team during the move. And this is Anna Ziknitsky-Sharp, who's the one who developed all of the male infection models. And I just want to acknowledge everyone in the lab and our funding, and certainly the core facilities that helped us really get at a lot of these questions. So with that, I am happy to open the discussion.
Thank you very much. That was a beautiful talk, Dr. Ingersoll. The slides were wonderful, and you described things so well that even someone like me who doesn't know much about immunology gets it. So I appreciate that. There's a couple of questions I want to get in the chat, and then people can raise their hands and we'll call on them. One question is from Pradeep Tyagi at University of Pittsburgh. He asks, is the T cell response upon macrophage depletion surprising? Mice simply clearing the bugs with whatever toolkit is available to them upon macrophage depletion. And then asks, are the mice females or males? Is there a difference there?
So the shift to a TH1 during macrophage depletion, is that the question?
It came earlier in the discussion, but yes, go ahead with that. Yeah.
I'm looking to see the timestamp to see if I can guess what slide I was at.
Yeah, like 20 minutes ago.
So I'm saying in general, I think clearance is really, clearance itself, I think is absolutely mediated by monocytes and neutrophils. I think we were surprised to see this shift in polarization, but what we know now with that TRM population, I'm not sure that means anything. We published the macrophage study before we did that last experiment for the T cell stuff. So it was at the time we were like, oh, of course, it's just TH1, and that's why we have this scenario. But now I'm not sure that's actually what's happening. So I think it's more surprising that we can take away cells like macrophages and we don't see an impact on bacterial clearance in a first infection, for example. So I think that was our most surprising finding in general. That macrophages and T cells are communicating, that's not surprising. And I'm really excited to keep going with these studies to understand exactly which ones are talking, where they're doing it, and what they're saying to each other.
Thank you. Another question is from Kathy Mendelson. She's wondering who makes the cytokines? Could it be epithelial cells?
So definitely, Kathy, I had this conversation today in my office with a new student. We have a new model where we do epithelial sheets, and the purpose of that is specific to know what cytokines the epithelial cells are making versus the cytokines that are coming from the macrophages. I think we're going to have to go a bit reductionist and really take the bladder apart to understand who's doing it, because it's a little bit more straightforward than trying to stain cytokines in situ, because this is hard, but that will ultimately have to happen, I think. But I think absolutely the epithelial cells are making a big contribution to the cytokine response.
Could I ask another question?
Yes, please.
Eosinophil, when I hear that, eosinophils, I thought of interstitial cystitis, which is another thing that males don't get as much as often as females. So you want to say something about that?
Yeah. Yeah, I love that. I don't know is the short answer, but I completely agree. You see those eosinophils and you think allergy, you think kind of, out of control inflammation, and certainly I see, right? So I would love to, and if anyone's working on this and wants to work with us, it'd be great. I'd really love to develop some sort of infectious insult model that ultimately leads to something that looks like IC. But I think that could be what they're doing. We've done some experiments where we've depleted them and it doesn't change anything. It doesn't change anything in terms of clearance, and it doesn't change anything in terms of CFU after a primary or second infection. So I don't think their first role is really a direct bacterial killing type role. So I don't know if they're helping to polarize macrophages towards a more healing phenotype, for example, you know, to repair after infection, or what they're doing there. But yeah, definitely their behavior gone awry. I would not be surprised to see a role for that in IC.
Let me just take 1 second to pull everybody's attention to the chat. We do have a link to a survey there that our CAIRIBU Interactions Core would like you to complete if you can. One will be emailed to you if we have your email address. There's a couple of other questions in the chat. One is, in a clinical setting, do you think during the RUTI infection in females is downregulated by Tregs or the absence of TRM cells in the bladder?
So I see who this is from. He's coming to Paris to help answer that question, which is fantastic. But that's really kind of where we are right now, right? So the question is, in a clinical setting, in an individual that keeps getting infection, is it because they didn't develop TRM cells? Is it because they developed them and they aren't sufficient? Is it because the Tregs are somehow suppressing their activity? We know there's a really nice study that is in the, I think it's in the lung, looking at the interaction between Tregs and TRM. And basically, Tregs help the development of TRM, if I'm remembering correctly. But ultimately, I think those are really open questions that we just don't know. And I think we need to first work out what's happening in the mouse, and then we can start asking those questions, probably through imaging in human bladders. It's a great question. I have no idea.
In the chat, also John Barrage notes that they have a mouse model that might be of interest. John, do you want to expand on that?
Yeah, I'm reading that too. I think that looks awesome. You know, sign me up. How does that mouse work? Yeah, so what we do, like all of our sequencing, we sort, and there's certainly, I think, really strong caveats to sorting cells. You know, you have to digest the tissue, and they're at 37 for 1 hour. When we digest, I like to say we use manual agitation, but what I mean by that is we shake the bladders really, really hard once every 15 minutes over a period of 1 hour. So it's harsh, right? And then the sorting itself is harsh. So I'm always curious what we're missing. So any opportunity to have information from specific cell types in a less abusive approach would be, I think, really, really important for us. I will say, anyone who's interested in this type of work, we love to collaborate. Now that we're allowed to fly again, we're happy to come over, but we also routinely host researchers who want to learn these methods in our lab in Paris. With croissant.
That's wonderful. Thank you so much, Dr. Ingersoll. This has been a really catalyzing discussion, and thank you for noting the collaborative spirit that you embody, because I think that's kind of where we're all headed, is more collaboration and more intersection of the different disciplines and the different fields and the different techniques that we all know. So thank you so much. Dr. Mendelsohn, would you like to say anything in conclusion? And then I'll give it to Dr. Ingersoll.
Well, if anyone is interested in using the male UTI model, we happen to have done it also. And I think John Barash's lab has done it. And I think Chad's lab has done it, Chad Vizina. So you guys, if you need to reach out to any of us for that, because that was a production, right? Getting that going. Yeah.
For sure.
Thank you. Dr. Ingersoll, any last words, key take-home points?
The bladder is the best organ. I mean, I don't know if that's controversial to say, but I would just say one of the best things about this field in general is it's small, it's friendly, and I feel like there's amazing number of questions that are outstanding that you know there's room for everybody.